Avans. Good afternoon, everybody. Welcome back. And this is the second lecture, continuing from the first lecture. I just want to note that this was we we developed this together. This is a very good friend of mine and a colleague, Marieke Bruins at Wageningen University. I just want to give her credit for the work that she did in making this presentation. So this lecture is actually a two-part lecture because, as you see, it's 61 slides. So we've split that up into two sections. This afternoon, we'll do the first half, and the assignment for that is assignment 2. And tomorrow morning, we will do the second half of biomass composition, and that will be an assignment 3. You will also notice that in assignments 2 and 3, that assignments 2 has no research question, because 2 and 3 are kind of put together, and the research question on biomass composition is put in assignment 3. But they should be completed separately and uploaded separately, so don't combine assignments. Biomass composition. So we talked about in the first lecture about you have different types. We saw maize, we saw sugar beet venosa, we saw lots of things. They had different dry weights, different crop yields, and they also had different composition. We divided then into fibers, proteins, lignin, and carbohydrates. And in this lecture, we're going to go into much more detail about what those are. Biomass composition. Before we fill this in, I just want to show you how we're going to approach it. So these are the same ones we talked about in the first lecture. You have leaves, grass, wood, algae, sugar beet, and sugar cane. The composition in general we'll be talking about are water, cellulose, hemicellulose, which is very similar to cellulose, lignin, starch and sugars. Starches are types of sugars, so just a complex sugar, protein, fat, and ash. We will be talking about the composition. This is all by dry weight, or dry. Sometimes it's called dry weight, sometimes it's called dry mass. Dry mass is actually more correct, but quite often it'll be called dry weight. I say dry weight and dry mass interchangeably, and most people do, but dry mass is technically better. And we will talk about a biorefinery type for this composition, appropriate for this composition. So water. Let's just leave. This very good question. This is for the whole plant, the sugar beet, the whole plant, and also for sugar cane. So here it says sugar beet, sugar cane. We are keeping it general though. Quite often it's very difficult to find data, and if you see something, so it includes the root. Yeah. The sugar beet is you can divide it actually into 3 parts. You call it the root, the stalks, and the leaves. But they have almost no stalk. They are mostly leaf. But yes, that's why it's very important. Again, I'm going to bore you by saying this so often. It's very important for you to do this analysis on your biomass when you get it in your hands, and only use data from books or online if you can't do it yourself. So water. Water is almost always the largest component of most biomass. The next part is lignocellulose. Lignocellulose includes the cellulose, the hemicellulose, they're slightly different, and the lignin. And quite often they're put together into one word, lignocellulose, which is it's like a jargon word for fiber. And we'll be talking about that. Lignocellulose, it's present in almost all plant material. It's part of the cell wall in cell plants, and it adds resistance to degradation and pathogens. So here we have the lignocellulose structure. This is the cell wall of a plant cell. The lignin is then the outside. It's like a cage. The hemicellulose is a wiry structure that goes through the cellulose, and then you have layers of cellulose through the cell wall. A slightly deeper inspection of the cell wall. You have the primary wall with the lamella. This is the primary wall. Then you have a secondary wall, followed by a plasma membrane. Notice the blue dots are the cellulose. The hemicellulose is threaded throughout, and there are proteins. You see this protein here, proteins that are throughout the cell. And then this is the inside of the cell, the nucleus of the cell, and that's where most of the water is. That's why the total mass has such a large percent of water, because the rest of the stuff we're talking about, the hemicellulose, is mostly in the cell wall. Examples of heavy lignocellulose feedstocks I mentioned yesterday in the kickoff. They include cereals such as straw, stover, and husks from, like for example, wheat. Sugarcane, that's a bagasse, tops and leaves. These are trash. A bagasse is leftovers. Woody biomass, here pictured here on the right. Energy crops such as miscanthus or switchgrass, and also oil palm biomass. Not the oil itself, but the empty fruit branch, the fibers, the shells, the trunks. So here you see the shells of the fruit of the oil palm. This could also include shells of any nut. Also the nuts of trees which are used for, let's say, the rubber tree, as I mentioned, I think yesterday. The rubber comes from the gum or the sap of the tree, and they still have lots of nuts that fall on the ground, and these can be picked up and used for lignocellulose feedstock. The composition of lignocellulose on a molecular level, we should understand it on a molecular level because it's important to know. Lignocellulose is a complex structure consisting of approximately 35 to 55% cellulose, and then cellulose itself is a polymer of glucose, a C6 sugar. So glucose, C6H12O6, that's the molecule definition. You can polymerize, put them chains to each other. I'm going the wrong way. Chain to each other to make a polymer. A single cell of glucose is called a monomer, and if you chain them on top, it's called a polymer. And that's what cellulose is. It's a polymer of glucose molecules. Hemicellulose, which is for most plants, again, this is all by dry mass, 25 to 45%. It's a polymer consisting of xylose, which is a C5 sugar, arabinose, galactose, certain acids and minor components. But again, it's mostly also a polymer of different sugars. The fact that these all end in OSE means these are sugars. Lignin, 20-30% by dry weight, by dry mass, is a complex molecule built up from phenolic alcohols. So cellulose and hemicellulose are very similar to each other structurally. Cellulose is a linear molecule, just one and another and another. Hemicellulose can have side structures, branches. It can have acetylation. So it's a slightly more complex polymer, but both of these are made up of sugars. Only hemicellulose uses different types of sugars. Lignin is very different than hemicellulose and cellulose, and as you see here, it's all types of phenolic acids. Here you go. There are, we'll go into more detail. I won't start right now. Okay, so here are all different types of lignocellulosic materials, from nutshells, wheat straw, leaves. Anybody out there in the sugar beet leaves territory? So if we look at the leaves, they should be approximately, by dry weight, 15 to 20% cellulose, 80 to 85% hemicellulose, and look at that, 0% lignin. That's why I was very careful when I said before that most cells have lignin, most plant cells have lignin, but not all of them. Leaves, in general, also do not have lignin. Grass does, the different type of waste used in cellulosic feedback. Oh, and I'll just one more thing. You may think this is weird. Swine waste, excuse me, is and cattle manure. I don't want to miss that too much. These are sometimes added to cellulosic biomass because of. Anybody have an idea? Why would we add manure to a biorefinery of lignocellulose? Cellulose. What's in manure? You have nutrients, which is also good to have. But you also, huh? Bacteria. Thank you very much. We got an answer in the room from Paul's. Bacteria. So if you are going to try to produce biogas, let's say methane, um, cow manure, swine manure has the bacteria, live bacteria, which can produce methane, but also CO2. So quite often it's added as a component. Normally, in like, I'll give you an example. Um, in some non-first, traditionally first world countries, they will produce their own natural gas for burning on the stove, methane, and it's popular to have an underground fermentation tank where all this they have to do is put in cow dung or whatever creatures they have around their farm, plus their waste from the rice fields, and it produces underground methane, which then they can use for cooking. It's not so common anymore in Europe or the United States, but for some of you who may find it interesting, gas lamps in London used to be actually fueled by the methane escaping from the sewers. That's just a little known fact. Until a few of them exploded, then they thought maybe health and safety should take over. So cellulose for chemists. As I mentioned, it's a very long straight chain of polymer of glucose. How long? It's approximately 10,000 glucoses in a row for wood. Cotton is nearly pure cellulose. Cellulose molecules link up in bundles and bundles of bundles and bundles of bundles and bundles to make fibers. So if you see, for example, cotton, which is almost pure cellulose, that's why cotton's so soft. It doesn't have much lignin in it. Doesn't have any lignin in it. It's a fibers upon fibers upon fibers, like what are those things called? The fiber optic cables. Cellulose forms tight bundles which are very resistant to chemical attack. Cellulose is pretty tough stuff. Hemicellulose, however, they're sugars, they're sugar polymers, but different from cellulose because they are made of glucose and other sugars. They do have glucose, but all the other sugars as well. They contain also non-sugars. They are branched polymers. They're much shorter polymers. They are about 50 to 300 polymers of 50 to 300 sugars instead of the other one was 10,000. Remember, in wood. And there are many, many, many different varieties of hemicelluloses depending on the structure of the hemicellulose and what non-sugars are there. It's less resistant to chemical attack, and it's easily broken down to simple sugars, hemicellulose, because it's not as tight. So it's 550 to 300 sugar units as a polymer of those 50 to 300 units. It composed of 5 carbon sugars, xylose, arabinose, 6 carbon sugars, mannose, galactose, and glucose, uronic acids, so it's galacturonic acid and glucuronic acid, and acetyl and methoxyl groups, such as acetic acid and methanol. Now, that was hemicellulose. We'll go on to the third and final part of the lignocellulose structure of plants. Lignin is a three-dimensional polymer. It's quite different than the other two, which are more linear. There are no sugars in it whatsoever. Lignin is sometimes called nature's glue, so it holds the cellulose and the hemicellulose together. Now this glue is actually for us somewhat of a problem because it keeps it in too well. If we want to release the proteins or the cellulose to make sugars, simple sugars, we have to destroy the lignin because it's such a good glue. It must be removed or weakened to separate the fibers and to turn into wood to pulp, for example. It's very dark in nature, especially after reacting with alkaline solutions. Let me, where did that go? So it must be decolored to remove from pulp. One of the industries that uses the most chemicals, certainly maybe the most bleach, is the paper industry, which is why recycling paper is so important. This is what the structure on a 2D structure of lignin looks like. We have lots of different connections, but it's not a 2D structure. It's more, can be described as a three-dimensional chicken wire. So here you have a chicken coop. You see this chicken wire? Now imagine this is just the chicken wire in 3 dimensions. Yeah, so you have like cubes upon cubes of the chicken wire, and that's what how lignin acts. It gives it the structure, of the cell wall. Now we go to carbohydrates. I mentioned yesterday about CHO molecules. That's what this is: carbon, hydrogen, oxygen. So carbohydrates are basic. Their basic chemical structure consists of sugar units. That means they contain carbon, hydrogen, and oxygen. Sometimes carbohydrates are referred to as CHOs, so carbon, oxygen, hydrogen. But normally, a chemist write that CHO. So sometimes you'll see CXHXOX, and that's the formula for a carbohydrate, such as C6H12O6 is glucose. Often shown as an aliphatic or linear structure, but exists in nature as ringed structures. So aliphatic means basically only containing CH and O, and linear meaning that they are connected to each other as a chain, linear chain. So here we are. This is a glucose perspective. Here is the linear structure I mentioned, and here is the ring structure of a sugar. This is glucose. Classification is made by the number of sugar units in the molecule. So as I mentioned before, we have monosaccharides. Glucose is one example of monosaccharides. Disaccharide, which has 2 monosaccharide units. So example is sucrose, is glucose plus fructose. You have polysaccharides, which are a long chain polymers of monosaccharides, and these are also called starches. The most important polysaccharide for biorefining are starch and cellulose. So if you hear starch, it is just a polysaccharide. It's a polymer of many different sugars. So here we have starch. As you see, it is combined in this way. The carbon from both rings is attached via a water molecule here in this configuration. And cellulose is very similar, but the bonding on the oxygen molecule is slightly different. It has a different orientation, the oxygen, between the 2 carbon atoms. That's the difference between starch and cellulose. Starch contains an alpha D glucose linkage. That's the name of the structure, the binding. Cellulose has a beta D glucose linkage. Starch is a storage sugar, so plants use it to store energy, such as potatoes and cassava, and it's easily degradable. Cellulose is the primary component of plant tissue and is largely indigestible to monogastrics. And what does a monogastric mean? That's yeah, we're monogastrics, but what does that mean? We have one something. Stomach, exactly. So monogastrics like us, we have one stomach. Give me an example of a non-monogastric. Oh, cows. Thank you. Very good, Yannick. Cows, but some monogastrics. Now we're going for the bonus round. Well, some monogastrics do can break down cellulose. Anybody give me an example of one of those? What eats lots? Monogastric eats lots and lots of grass, for example. Hmm? No, cows are not monogastric. They have more than one. They have several stomachs. Uh, yeah, so goats, elephants, gorillas, those are examples. They have that's why gorillas have such very large bellies. They eat lots and lots of grass. Pandas, yeah, pandas also. But by and large, I'll go back. By and large, most monogastrics cannot eat grass directly. It's too low energy level and too much cellulose. Must have enzymes to break it down. So we need enzymes to break it down. All enzymes end in ASE. So a cellulase breaks down cellulose. That's a very quick. We'll talk about enzymes later, but that's an easy way you can tell if something is an enzyme because they put ASE instead of OSE. And you can also tell what it breaks down. So cellulase breaks down cellulose. Um, Glucase brings us to glucose. Protease breaks down proteins. There you go. Now you've learned it a small little bit. What is starch? So it's granules extracted from potato, corn, wheat, tapioca, rice. Serves as storage of carbohydrates. We mentioned that already. It depends on its origin. So it can be modified starches within it. Not all starches are particularly the same. They are insoluble in water at temperatures below 50 degrees Celsius, which is why normally if you eat starchy products, you normally eat them at they have to be uh, they have to be brought to a high temperature. Right. You boil your rice in hot water. Um, Otherwise, it takes a very long time to eat it, so it has to be broken down. It has a higher density than water, so it can be extracted by sedimentation or centrifugation. So, if you wanted to get starch out of rice, you could just boil the rice, dissolve the starch in the water, and then just cool it down below 50 degrees Celsius, and just centrifuge, and you've gotten rid of the starch. In plants, they're found specifically in seeds, bulbs, and tubers. So these are the things that we eat. Actually, we can eat get use starches quite well. They plants which are high in starch will not surprise you. Potatoes, which are 20% starch, rice, which is 80% starch. A general, if you have a combination, is 75%, and oats, which is about 2/3 starch. This is what starch looks like under a microscope. I think this is a scanning electron microscope. They are there's polarization. Slightly, what is starch more specifically? It's an alpha 1,4. This is amylose. so it's mostly linear. So what does that mean, 1,4, alpha 1,4? Alpha 1,4, the alpha means how the linkage is between each other, and the 1,4 means the first carbon, 1, is connected to the 4 carbon of the next one. See? The carbon, so this is the 1 connected to the 4. So we count from here, from left to right. So 1, 2, 3, 4. This is connected to the 1, 2, 3, 4, connected to the 1. So you see that? That's why it's a alpha 1,4, an alpha link on the first and the fourth carbon polymer. A 1,6 linked glucose unit is called amylopectin. These are all starches, by the way. Amylopectin, this is a 1,6. Here's the 6. Actually, kind of the wrong way. Oops. So this is 1, 2, 3, 4, 5, 6. I went clockwise. You should go counterclockwise. So here's 1, 2, 3, 4, 5, 6. So this is connected a 1,6. Regular starches consist of two polymers, approximately 20% amylose. Yeah. Oh, good question. Yes. Yeah. Because of the connection here. We count, well, let's start here. So this is the 1 carbon, and this is the 4 carbon. So this is 1, 2, 3, 4 connected to the 1 of the next one. So it's the 4 connected to the 1, connected to the 4, connected to the 1, connected to the. You see? These are the two main starches, by the way, as I mentioned in the next one. So you have amylose, which is almost linear, and you have amylopectin, which is branched. And it's this ratio which is interesting. Yeah. Uh, so there are different types of potatoes, right? If you go to the grocery store, you'll see different types of potatoes. And some of the potatoes will say are for cooking, and some potatoes are made for like mashing. And the reason why they are different is because of their starch. The normal potato will be about 20% amylose and 80% amylopectin, so 20% linear and 80% amylopectin. And a waxy potato is 3% amylose and 97% amylopectin. So not only is the percentage of starch important, but the division between amylose and amylopectin. Can anybody think why this is important to know? What kinds of starches are in a potato, for example, or a rice? Yeah, breaking it down, exactly. So the starch, um, if it's high in amylose, it's much easier to break it down. You can just use a simple enzyme, an amylase, and you'll have in no time simple sugars, which you can add to lots of things, to a fermentation technology, for example. If it has a lot of amylopectin, it'll be much more difficult to break down because it's very branched. It's not just sugars. It's more difficult to get to biologically. Sources of starch. You have a couple of different types of corn. That's why some corn we call it animal corn because of the amount of amylopectin. So maize or corn. You also have waxy maize, waxy corn. You have potatoes, normal potatoes, and you have waxy potatoes. Tapioca is made from cassava, which has to be broken down. Tapioca, if you eat tapioca, which I actually find delicious. It's from the cassava, but needs to be processed first. Wheat, the same, but peas, uh, rice and waxy rice, they all have different starch raw materials due to its composition of amylose, amylopectin, granular size, and other minor constituents and water concentrations. Okay. Um, I'm just wondering how much further I want to go. I've been talking for about a half an hour now. Um, amylopectin is also branched. So, how many normal normal chains of glucose to a branched is very important. And amylopectin, you have different types of amylopectin. So you have A, B1, B2, C. Um, there's, hmm? Oh, uh-oh. Is it okay? Are you doing well, or do you need help? Just follow on the front. Okay, there we go. I'm going to. I think I'm going to. I'll continue just a few more sheets, and we'll finish this at Starch, and I'll let you go on to your homework, your assignments. So there are different levels of starch, uh, similar to. Is it going? Similar to other biological materials like cellulose. It's we don't you don't. So hemicellulose is very linear, very long chains, bundles and bundles of cellulose. And starch is a little bit like that. It's starch upon starch upon starch upon starch upon starch, and it builds up. So if you were to cut through a single starch grain or granule, you'd see all of these layers. So it's all wrapped up on top of each other and wrapped up, wrapped up, wrapped up. So it's a starch structure is semi-crystalline with like growth rings. Oh, it's very similar to trees actually. If you can cut through diagonally, have a somewhat amorphous background, and then once you get into much smaller, there's about a 9 nanometer difference between them, and within that crystalline structure, you have the chains itself. So here you go, unraveling it from, this is at 10 micrometers, zoomed in from 1 micrometer, and even further zoomed in at a level of 200 nanometers. If you go even further zoomed in, you see a spiral structure. And then lastly, here we're talking at the molecular level. So you have the polymer wrapped together in spirals, the spirals themselves bundled into sheaths, and the sheaths layered upon layered upon layered, and then it can fit in to the cell structure. And here you see, the lignin. Remember I said it looked like 3D chicken wire? I wasn't kidding. This is the 3D chicken wire lignin part. If it's a very fibrous root, like like a carrot, it'll have a lot of lignin. If it's a less fibrous root, like a potato, it'll have much less lignin. I think we're just about finished. So here we have a few different crops and their percentage of amylose, amylopectin, and water. Yeah, so all between about 13 and 19%. But a waxy potato is virtually 100%, going down to corn, which is about 28% amylose, which is quite often why corn is used as a sugar to make sugar. Um, Here's the percent of, again, dry mass for proteins and lipids of these same plants. Maybe good for your homework, so for your project. And lastly, we'll talk about the degradation of starch before I end this lecture. It's Starch is actually really easy to break down. That's why we like to use starchy, high amylose biomass for sugar production. You can do two base ones are acid, so using sulfuric acid or hydrochloric acid or any kind of or biological acid. Will break down. It'll cut the what are those? Those are alpha one. It's alpha 1,4 linkage. Yeah, alpha 1,4. Remember 1, 2, 3, 4. Alpha 1,4. Also, you can use enzymes. The couple of problems with using enzymes. Yeah, maybe somebody knows. Does anybody know like why quite often we use acids and not enzymes? Maybe even take a guess. You guys have a guess about why we might use an enzyme instead of an acid? Well, no, we'd rather use an acid than an enzyme. And well, no, enzymes are just more. Yeah, there you go, Leonard. Got it. Yes, Leonard for the win. Cheaper. Yay! Acids are cheaper. Enzymes are expensive. Enzymes, as we'll talk in I think tomorrow, maybe a couple times even enzymes, definitely tomorrow. Enzymes are expensive. Quite often they're designer enzymes, so you have to produce them. Enzymes, as you'll probably already know, are not affected by the reaction themselves, but they're impossible to recover. Not completely impossible, but very, very expensive to recover your enzymes. So once you use the enzyme, even though in a chemical way, in a chemical process, enzymes are not consumed. However, in the real world where we have a refinery, you empty your enzymes and you basically, you're using it up because it just goes into your product. Of course, the enzymes are so small that they don't harm your end product at all, but they're expensive and they are you can't get them back unless you put a lot of energy into getting the enzymes back. It's just cheaper to buy more enzymes, and an acid is much cheaper than enzymes. So that's why quite often acid is used rather than enzymes. Okay, that's it. Yeah, I'll stop there. We won't go into proteins this afternoon. Before I let you send you off to do your second assignment this afternoon, are there any questions for me? Anything? Okay. All right. We'll do that after then. Anything specific about the lecture, people? I can. How about I do a short review because we talked. We've talked. I talked quite a bit of time now for 37 minutes and 57 seconds. So quick short review. Okay, we looked at what is dry matter again. What is lignocellulose? Lignocellulose are three things: lignin, cellulose, and hemicellulose. What's the difference between cellulose and hemicellulose? Cellulose is linear, hemicellulose is branched. We looked at starch, different types of sugars. So a starch is a polysaccharide, so it has many sugars, more than one in a chain. A disaccharide is a double, and a monosaccharide like glucose is a single. And we talked about the different types of starches and how that's bundled together within a lignin structure. And lastly, how we can differentiate different plants in their biodiversity through their carbohydrates and their starch contents, as well as protein, etc. Okay, I'm going to stop the recording. If anybody has questions, I'll be here until what time do we have? Do you remember? Like around, yeah. So 3:30. I'll be here until 3:30 to answer any questions on the assignments. Otherwise, I'll see you guys tomorrow morning. Bye bye. Oh, don't forget the assignments are due before the next lecture, both 1 and 2.